Hello, and welcome to your presentation on homeostasis and cell transport. There are two main things that you will need to know. First, the cell membranes are selectively permeable due to their structure. Selectively permeable means that some things are able to pass through, but others are not. And what determines what can go through and not is because of the structure of the cell membrane. Also, homeostasis is maintained by the constant movement of molecules across the membrane. Homeostasis is maintaining stable internal conditions. This includes such things as maintaining a steady body temperature. You shiver when you're cold to try and get warmer. It also includes making sure the right things are inside of your cells and outside. That's what the rest of this presentation will be about. We have two types of transport across our cells. There is passive transport and active. Passive transport is going to be when we move things from high concentration to low. So in other words, where there's a lot of stuff to where there's less. Think about it as just how things move normally. Active transport is going to require ATP to push things against the concentration gradient. So passive transport does not require energy, and it moves things with the concentration gradient. There are three main examples of this that we will talk about in a few minutes: osmosis, diffusion, and facilitated transport. And then active transport is the opposite, so it requires ATP. It moves things against the concentration gradient. The three main examples we have for this one are going to be protein pumps, endocytosis, and exocytosis. First, we have passive transport. The first example that we want to talk about is osmosis, which is the diffusion of water. If you remember that the formula for water is H2O, it helps you to remember that osmosis with an O goes with H2O. With osmosis and all diffusion, things are going to move from a high to a low concentration. Water will move through protein channels called aquaporins. This means this is a protein channel in your cell membrane where only water can pass. Okay, let's look at a picture of this and then we'll get more detailed. So we're looking at an example of your red blood cells. Your red blood cells normally have this little structure with a little dimple in them. Normally, water is moving back and forth, so there's the same amount of water on the inside or outside. However, if there is a higher concentration of water on the outside, water will diffuse in. Then look at this cell. That's not normal. However, if there is less water on the outside, so maybe you're dehydrated, water is going to move out, and again, that cell doesn't look normal. Now, another example of this, other than being dehydrated, is maybe if you had high blood sugar or low blood sugar. This is common with people who have diabetes and not able to control their blood sugar levels. You may have heard of these terms before. So, in this case, high blood sugar would be hypoglycemia, and because there's more sugar in your blood, there's a lower concentration of water. And then over here, this would be hyperglycemia, where there is less sugar or other solutes in your blood, so there is going to be a higher concentration of water. Okay, osmosis in animal cells. There's no cell wall. This means that if too much water moves in, the cell could actually burst in what we call an hypotonic solution. Normal animal cells are isotonic, so there's the same amount of water inside and out so they can maintain their proper structure. Here's a cool picture of showing what your red blood cells actually look like under a microscope. In this one, the middle one is normal. This one has too much water. If it continues to gain water, it could burst. And over here, the cell is crenulated or shrunk up because there's not enough water. Okay, yet again, another example of passive transport looking at how the cells are going to change in size. So if it's in a hypotonic solution, water moves in, the red blood cells increase in size. Isotonic is normal, water's going back and forth, no change in size. Hypertonic solution, water moves out and it shrinks up. And remember, go ahead and write this down, water always moves to hypertonic areas. Okay, short little thing about plant cells. Since plant cells have a cell wall, they won't burst if they're in a hypotonic solution. That's actually the normal condition for a plant cell. If a plant is wilting, it's because it doesn't have enough water inside of its cells to support the whole plant body. Okay, now let's get into the details of what we mean about all these tonic words. If a cell is in an isotonic solution, this means there is an equal concentration of solutes and water on the inside and outside. Let's use these arrows to show the movement of water. Water will be moving out of the cell and it will be moving in the cell, except for my arrows are not working right. Sorry. This means that maybe there is a 90% concentration of water on the inside. There's also 90% concentration on the outside. Again, if we're talking about an animal cell, this is what normally should happen. Now, what if we have a higher concentration of water on the outside? Wherever there is more H2O, it is hypo. So there's a hypotonic solution. That means that there's more water on the outside, so water is going to move in to the cell. If there is more water on the outside, the solution is hypotonic. And guess what? The solution on the inside is hypertonic. That means more water on the outside, less water on the inside. Okay, and then we get to a hypertonic solution. This means the solution that is around this cell has a lower concentration of water, so water will go from the inside to the outside of the cell. So in this case, water is moving out from a high concentration, so there's a lot of H2O in this hypotonic solution, to the outside where it is hypertonic. Okay, that was the largest bit of information. The rest goes a little bit easier. So number 2, we're still talking about passive transport, by the way. This is facilitated diffusion. In this case, we're going to have a protein channel that helps to move things across the cell membrane. It is still passive, so we're moving with the concentration gradient from an area of high to low concentration. Transport proteins are these purple blobs that are shown here and here, and they are actually very specific. This means that if you have a water channel, it will only transport water. If you have a sodium ion channel, it will only transport sodium ion. Okay, now to active transport. Make sure you note in your notes that active transport is separate than passive. If we're talking about active transport, the key ideas is that we're moving against the concentration gradient and we're using energy. The first example is an example of a protein pump called a sodium-potassium pump. The animation below is showing how that works. What we also want to understand is that sodium-potassium pump is one of the reasons that we need sodium and potassium, and we need this in order for our nerves to function. And then we have endocytosis. Endo means in, cyto means cell. So this is going to be taking in large stuff. There are 3 types. The penocytosis is cellular drinking. This is when the cell would take in large amounts of volume or of liquid. This is important in fetal development. This is how the cells get larger. Receptor mediated is going to use certain receptors to take in specific substances. That's what the animation below is showing, as those receptors on the outside of the cell membrane bind to this vesicle, they're going to take it in. If these pieces didn't fit together, it would not go in. Okay, and then we have cellular eating, which is phagocytosis. This would be how cells will eat large particles. There are two pictures shown at the top. One on the left is an amoeba about to eat some food, and then on the right is a white blood cell eating a yeast. And then at the bottom, there's one of your white blood cells chasing a bacteria. And it eats it. Okay, then exocytosis. Exo means out, cyto means cell, so exocytosis will be getting stuff out of the cell. A common example of this is going to be protein export from the Golgi. So we learned that ribosomes make proteins. The ER can modify those proteins, send them to the Golgi. The Golgi will further modify them and package them for export. And the way they are exported is that vesicle will fuse with the cell membrane and release its contents. Okay, now that you have learned about the cell membrane and homeostasis and cell transport, you should be able to explain these two statements.